I'm Dr. Teresa Lyons, creator of Navigating Autism and Eat to Heal Autism. And this week's Ask Dr. Lyons question is, what common biological pathways are disrupted due to genetic changes with autism? Autism and genes. So if you haven't listened to my other videos on genes and autism, please do. There's some great information. And if you have, then you certainly know this scientific fact. It is estimated that about 1,000 genes are involved in autism, which means that no one gene is likely to explain more than 1% of cases. Scientists have then tried to understand if there are common biological pathways that are disrupted due to genetic changes with autism. And that's what we're going to discuss today. We're going to look at nonsense mutations. And in the previous video, I went over what genetic changes actually mean, what these mutations actually do, how they change things. So if you're not familiar with nonsense mutations, just pop on over to the previous video and take a listen. Nonsense mutations, just to summarize, cause the resulting protein to be truncated, or also known as being shortened or incomplete. So you have a stop codon that gets produced with a nonsense mutation. So these kinds of mutations cause a very noticeable change in the protein. If the protein gets shortened or truncated, then it's a much bigger change and easier to study in a way. Scientists studied this large genetic change since it would be easier to understand the effect and determine if there are common biological processes involved. One study revealed that the beta-catenin chromatin remodeling protein network was involved. So let's dive in a little deeper into that. So beta-catenin. Beta-catenin is a multifunctional protein that contributes to cell development, cell regeneration, and cell-cell adhesion. So I'm just going to explain the role. Next slide, I will show you a figure so that it can all come together. So in unstimulated cells, beta-catenin resides in the cytoplasm, and it's not allowed to build up in the body, in these cells. It's constantly targeted for phosphorylation and degradation. So when the body needs beta-catenin to be involved, it gets stimulated. And it gets stimulated by specific signaling pathways, one of them being the WNT ligands. And the beta-catenin protein is then stabilized and it shuttles, basically it moves from one part of the cell to another part of the cell where it is more involved in transcription. And there's some specifics of how it's involved in transcription. It associates with coactivators such as the T cell factor, lymphoid enhancer factor, family members, and through all these different interactions, that's what activates gene transcription in this particular case. All right, and here's the figure that I promised you. So on the right-hand side, that's a cell. And you can see, let me just get my mouse here. There's degradation going on when B-catenin is in this area of the cell. When it's needed, it gets stimulated and goes into this area of the cell. And this is where it gets involved in transcription. There's a lot of other things going on in this figure, but I just wanted you to get the overall feel for what beta-catenin is involved in. Chromatin is DNA and protein that forms chromosomes. So that's also involved in this beta-catenin process. So now here's the slide that we saw before that just uses words to describe everything. And I'm sure it's a lot clearer now. And again, the important thing to remember is that beta-catenin is involved in cell development, cell regeneration, and really importantly, cell-cell adhesion. Other mutations and processes that involved. It's not just one process that's involved. So there were 12 other de novo mutations that have been studied by various scientists and found with high reliability certain common biological processes are involved. And those are neuron cell-cell adhesion. So again, cell-cell adhesion is something that keeps coming up as genetically important for autism. Vocalization behavior, glutamate receptor signaling pathway, cognition, and neuron projection. Other processes. So in addition, other genetic studies have found that these biological processes are involved as well. Synaptic gene regulation, actin-skeleton dynamics, cell growth, and calcium signaling. So what good is genetics, right? These are some kind of large biological processes. So again, it's estimated that about 1,000 genes are involved in autism, which means that no one gene is likely to explain more than 1% of cases. Genetics is complex. I'm sure you realize that. But thankfully, there are common biological processes. And this is how physicians are taking what is known in the lab, studying genetics, and translating that to helping your child. If you like this video, please give it a thumbs up. You can also subscribe to my channel so that you get notified as soon as I post a video. And here are some references.